Hello, all. I am Eric Brown, and I'm going to share some progress on some work I've been doing on Bornean squirrel temperature regulation physiology. At this point, I would expect that a lot of you are familiar with endothermy and my general interest in mammalian regulation of body temperature and metabolic rate, but a refresher for those who don't. Here is just a spectrum of the different ways that mammals are known to regulate body temperature and metabolic rate, which can be very flexible, as in the instance of hibernation and various forms of torpor, which we call heterothermy, or it can be very strict control of body temperature and metabolic rate, which we term homeothermy, which is very familiar to us as humans because that is what we do. Importantly, each of these ways to regulate body temperature and metabolic rate has an associated energetic cost. And in the bottom panel of this figure here, where T and Z is, this is the thermoneutral zone, denoting a range of ambient temperatures across which metabolic rate does not have to be elevated to maintain a preferred body temperature, but beyond which there is an associated cost of elevated metabolic rate to maintain that body temperature. And for each of these different colored lines, there is a different profile of energetic cost, the black being normothermy or homeothermy, basically keeping body temperature at a particular range across ambient temperature regimes. So this one is highly costly. The blue denoting some flexibility in body temperature to track ambient temperature to a certain degree, lowering the energetic cost, and then orange and red denoting various forms of torpor or hibernation, which includes active suppression of metabolism and oftentimes also a decrease in body temperature. And I would assume at this point that you are all Team Squirrel as being the model for studying thermoregulatory physiology in mammals, and that is because they show all of these different kinds of ways to regulate body temperature and metabolism, and they are also pretty much everywhere. You can find them in most places in the world, and there are a lot of different kinds of them. Now, it's all well and good, but historically, a lot of these studies have been biased towards higher latitudes, especially in the Nearctic or North America, parts of Europe and Northern Asia, and we have quite an absence of understanding about the species that live in tropical and subtropical latitudes. Likewise, there's also quite an absence of information about the phylogenetic diversity of squirrels and a bias towards the marmotine squirrels, or what we might know as marmots and ground squirrels, chipmunks, prairie dogs, and the like, as well as a couple studies on North American tree squirrels, flying squirrels, and just a handful of African ground squirrels. But notably, there is a very large gap, especially in Southeast Asia and South America and most of Africa, where there is still quite a bit of diversity of squirrels. And so I, in my project, have attempted to fill in some of the gaps with a few species coming from different genera from the Indo-Malayan region. And so the first species is Calliceratus notatus, or the plantain squirrel, the three-line ground squirrel, Loriscus insignis, and low squirrel, or Sunda lowii. And in this project, I have attempted to determine how being in a very unseasonal environment affects the thermophysiology of these squirrels as compared to many of the studies that we have historically, which focuses on squirrel species in very seasonal, temperate areas. And so we expect that for these species, which are varied in their size and zone of activity in the forest canopy, that they will lessen their energetic costs by allowing their body temperature to a certain degree be flexible with ambient temperature. And so we went into community forests in western Sarawak, Malaysia, on the island of Borneo in Southeast Asia, which is kind of all in the Malay archipelago. And we trapped squirrels. We took some various morphometric measurements and ultimately measured metabolic rate, or at least proxies for metabolic rate, with a method called flow-through respirometry. So we take these individuals, we put them in a box that goes in another box for which we can control the temperature, and then we flow dried air into the box with the squirrel, and then that shunts out air into a gas analyzer where we can measure the oxygen consumed by the squirrel, the CO2 and water produced, and we can get a sense for the metabolic response to a particular temperature regime. And importantly, individuals that we use for respirometry are always adults. They are healthy as far as we can tell, and they have not recently eaten. And if they are female, they are not visibly pregnant or lactating, and they're also always at rest. We're looking for a measure called resting metabolic rate, which is kind of a standard comparative measurement for respirometry. And our setup is very high tech, very expensive. I mean, it is very expensive, but being a little facetious with the fanciness here, our nice little Coleman cooler there. But inside we've got our guest, which oftentimes looks like this annoyed female, which eventually becomes resigned to their fate and chills out and goes to sleep through the entirety of the trial, which typically runs from dark to light, so 6 or 7 p.m. until 6 or 7 a.m. during their rest phase. And then we can get a sense of how body temperature reacts to these different phases, active phase versus at rest. And during the beginning of these trials, as you can see in these 3 panels reflecting each of the 3 species, that as they kind of come out of their active phase around 6, 7, 8, 9 o'clock in the evening, they have an elevated temperature that kind of dips through the night. And that remains lowered until they begin to wake up or prepare to wake up for the day around 6 or 7 a.m. and body temperature starts to recover. And boiled down to just one temperature trial per data point here at a given specific ambient temperature challenge. I'll focus just on the black dots here, which is the largest squirrel, the plantain squirrel. And forgive me for not having my regression lines ready to go, but we can kind of see that there is an increase in body temperature associated with increases in ambient temperature. And as we look at resting metabolic rate here, or metabolism, this is a proxy for metabolism, that at lower ambient temperatures, metabolic rate increases, but at higher ambient temperatures, metabolic rate is lowered. And the significance between having elevated body temperature at higher ambient temperatures but lower ambient temperatures suggests that there's some allowance for body temperature to increase to allow metabolic rate to remain lower. And then we also see that probably these squirrels aren't sweating much to maintain a body temperature at a particular level, because across the range of the temperatures that we challenged them with, their evaporative water loss, EWL, does not necessarily increase, or there doesn't appear to be a trend there. When we look at a phylogeny of species for which there is metabolic information available, we see that there is generally a lower mass-corrected metabolic rate associated with hibernation, or a species with these black dots here in this figure. except among food caching species such as various species of chipmunk. But besides that, there aren't really any clear associations between kind of regulation of metabolism or body temperature and the intensity of metabolic rate. And likewise for body temperature, we see, especially among some fairly recent marmot groups such as Cynomus or the prairie dogs, that there's a cooler general normal body temperature associated with these species that hibernate. But across the other species, that there aren't really any clear associations between kind of regulation of body temperature and metabolic rate and the degree of body temperature that they can achieve. And for both of these figures, I'll just say this palm tree denotes the species that we investigated. And so, even relatively minor flexibility in body temperature can confer considerable savings in energy. And now we can get a sense that the caloocyarid squirrels, or the beautiful squirrels, the three of which we investigated, also take advantage of the flexibility in body temperature to save some energy in a place where it is fairly routinely warm year-round. And we can get a better sense that this flexibility in thermoregulation appears fairly broadly represented throughout the squirrel phylogeny. And with that, I'd like to thank some friends and lab mates, Danielle Levesque, NSF, Climate Change Institute, UNIMAS, and my union for all the help in getting this work done and continuing the work.